Hello everyone, I am Dr. Meena Zore and Professor Siram Ramakrishna team members from National University of Singapore. I will present in behalf of Professor Siram Ramakrishna on the topic of current progress of Electrospun Nanocarriers for drug delivery applications. Professor Siram Ramakrishna recognized the world's top 3 biomedical engineers by Stanford University study. I am glad to participate in the first International Electronic Conference on Pharmaceutics. You are all welcome to contact us at the email address given in this slide. Professor Siram Ramakrishna published books on electrospinning and nanofibers. Few of his publications are including Electrospinning and Nanofiber Composite for Biomedical Application, An Introduction to Electrospinning and Nanofiber. Also, he wrote a book on Filtering Media by Electrospinning and Fundamental and Application of Micro Nanofibers. Also, he wrote a book on melt electrospinning. Nanotechnology is a rapidly emerging cross-discipline program that manipulates assorted synthetic and naturally occurring materials in nanoscale dimension. The advance of nanotechnology platforms has modernized several branches of science and engineering and contributes to societies by impacting renewable energy, energy storage, electronics, the oil and gas industry, water and air filtration, military applications, design and production of cosmetics, agriculture, the food industry, healthcare, the pharmaceutical industry, and the fast-moving consumer goods industry. There are two main approaches for synthesis of nanomaterials: top-down and bottom-up approach. In top-down approach, the nanomaterial will fabricate from bulk materials. These techniques are including spinning, sputtering, chemical etching, thermal or laser ablation, mechanical and ball milling, explosion process. And in the bottom-up approach, from the atomic and the molecular level of the materials, the nuclei will form and then fabricate and synthesize nanomaterials. This approach, including atomic molecular condensation, vapor deposition, sol gel process, spray pyrolysis, chemical electrochemical deposition, aerosol process, and bioretardation. Several methods are available for the fabrication of nanofibers and include electrospinning to produce random, aligned, torsional, and vertical nanofibers. Also electrospinning, rotary jet spinning, self-assembly, sol gel methods, phase separation, melt bowling protocol, and template synthesis. Electrospinning suppresses the other methods in relation to efficiency and has emerged as the most efficacious techniques for nanofiber production. The electrospinning process is continuous, scalable, and applicable to many different polymers and yield excellent control over nanofiber dimension and orientation. Electrospin and Electrospray Method has different applications in healthcare, including tissue engineering, drug delivery, food industry, agriculture, and cosmetics. It has application in water and air filtration and defense. Also in energy, including solar cell, battery, fuel cell, electronic, oil, and gas. Electrospinning is a beneficial and effective technology to produce continuous fiber by electric force. According to mechanism of the electrospinning process, the basic electrospinning setup contains a high voltage system, a spinner, and collector. To control the Electrospun fiber structure and for practical application, great effort have been made to modify the electrospinning setup and procedure. Based on the properties of the obtained nanofiber, there are several applications in healthcare and biomedicals. Tissue engineering, regenerative medicine, drug and therapeutic agent delivery, biological sensing, and wound dressing are the application of the nanofiber in healthcare and biomedical. Types of electrospinning, different therapeutics loaded nanofiber, and their route of administrations. Drugs and therapeutic agents such as antibodies, RNA, DNA, protein, hormones, and drugs can be administered to any region or organ in the body by the way of common routes such as oral, parenteral, sublingual, buccal, rectal, vaginal, ocular, nasal, inhalation, and transdermal using electrospin nanofibers. Nanofibers can obtain by different types of electrospinning such as blend, coaxial, emulsion, melt, gas jet electrospinning. In future, we will go into the detail of each electrospinning methods. Drug loading strategies to nanofiber is classified to three systems. The first one is basic antibacterial delivery systems. Second, advanced antibacterial delivery systems, core shell structure, nanoparticle, degraded, and multi-drug loaded. And the third one is smart delivery system, stimuli response. In the traditional and basic strategy of nanofiber-based drug delivery systems, the electrospinning process fabricates antibiotic-loaded nanofiber scaffolds either by making a solution of drug or nanofiber before electrospinning or by loading the antibiotic onto the large surface of the nanofiber through physical or chemical modifications. The advanced drug delivery system is for controlling the antibacterial drug release pattern from the nanofiber-based carrier. In this technique, the drug molecules are encapsulated into the core region of this advanced core shell structure of electrospun nanofibers, whereas the outer shells act as a protector and also manage the release behavior of incorporated antibacterial drugs. In the smart antibiotic delivery system, electrospun nanofibers scaffolds are commonly used to control the release of drugs in response to various biological parameters including pH factors, temperature, or UV light sensitivity. The biopharmaceutical classification system is a scientific framework for classifying drug substances based on their aqueous solubility and intestinal permeability. When combined with the solution of the drug product, the biopharmaceutical classification system takes into account three major factors that govern the rate and extent of drug absorption from immediate release solid oral dosage form. 1. Dissolution, 2nd, solubility, and 3rd, the intestinal permeability. According to the biopharmaceutical classification system, drug substances are classified as four classes. Class 1, high soluble, high permeable. Class 2, high soluble, high permeable. Class 3, high soluble, low permeable. And class 4 is low soluble and low permeable. Electrospun nanofibers for the delivery of active drugs through nasal, oral, and vaginal mucosa. According to the route of administration, the properties of drugs differ. For instance, properties of drugs loaded nanofiber in transmucosal delivery should have high mucoadhesive, stable formulation, high surface area, suitable for a wide range of drugs, tailored drug release, high drug payloaded, easy of administration via different mucosal tissues, biodegradable, and can be prepared aseptically. The drug can load it into the matrix of nanofibers, or drug can functionalize nanofibers, or drug can be in the core of the nanofibers. The retardation rate and dissolution rate of polymer is very important for selection of polymers for different administration routes. The main drug delivery challenges are solubility, degradation, and target delivery. By crystal modification, pH modification, size reduction, crystallization, solvent polarity adjustment, use of surfactants or lipids, use of complexing agents, drug dispersion can solve the solubility problem to some extent. The degradation of drug carriers can be modified by chemical degradation techniques such as oxidation, isomerization, hydrolysis, photolysis, and polymerization process. Physical degradation are including amorphous to crystal transformation and precipitation. Targeting drug delivery allow targeted deposition at the intended site, sustained release, safety, reduced dosing frequency, and patient convenience. There are two target delivery approaches, include passive targeting and second one is active targeting. Active targeting includes targeting mediated by external stimuli and antibody-directed enzyme prodrug therapy, and the third one is targeting in gene therapy. To have the effective drug delivery, we can modify solubility, degradability, and target delivery of the drugs. Nanocarriers for drug delivery approach and their biophysiochemical properties. Many factors are involving in the therapeutic potential of nanocarriers in drug delivery applications, including size, shape, materials, and surface chemistry of nanomaterials. The size of nanomaterials can be in the range of 1 to 100 nanometers and with the structure such as dendrimer, liposomes, polymer, hydrogel, metal nanoparticles. The shape of nanoparticles can be sphere, plate, cube, star, and rod nanoparticles as well. The surface functional group, coating surface charge, and targeted ligands impact on the properties of drug delivery. Various mechanisms for drug release from nanocarriers are including diffusion control release, solvent control release, polymer degraded release, and pH sensitive release. Diffusion control drug release occurs in capsule-like systems where the drug is dissolved or dispersed in core. Solvent control release. In this release method, transport of solvent into drug delivery systems may affect drug release behavior from the delivery carriers. Solvent control release includes osmotic and swelling control release. In degradation control release, drug carriers composed of biodegradable polymers such as polyester, polyamide, and polysaccharide release the drug from enzymatic decomposition, which degrades ester or amide bonds or cause hydrolysis. pH control release, the release of drugs from carriers that response to stimuli is controlled by a stimulus such as temperature, pH, ionic strength, ultrasound electricity, or magnetic fields. Such carriers have been studied for targeted specific drug delivery because it is possible to localize simulation. Here we can see solvent control release mechanisms for nanocarriers. The first mechanism is diffusion through insoluble polymer shell. Second one is diffusion through an insoluble polymer matrix, and the third one is erosion of polymer matrix. There are several spinning methods such as electrospinning, electrospray, microfluid spinning, solution blow spinning, and centrifugal spinning, and multi-spinning. Electrospinning and electrospray techniques are electrodynamic atomization methods. Electrospray approach fabricates monodisperse and fine aerosol of charged particles, while electrospinning prepares micro-nanofibers. Microfluid spinning is the method for generation of complex nanostructure and allow the accurate manipulation of fluid within microscale channels and allow for fast prototyping and bulk production of nozzles. The centrifugal spinning technique applies at high temperatures around 900 to 1000 degrees centigrade to maintain liquid state and spinning heat rotation more than 3000 rpm. Solution below spinning technique is applying a gas flow focusing liquid in low voltage to generate fiber using volatile solvent and pressurized gas that follow around polymer solutions. Types of electrospinnings for fabrication of nanocarriers for drug delivery applications. There are several electrospinning methods for incorporation of drugs for drug delivery applications, including solution electrospinning, emulsion electrospinning, surface modification electrospinning, side-by-side electrospinning, or genus multi-jet electrospinning, multi-axial electrospinning, coaxial electrospinning, and blend electrospinning. As you can see here in the figures, what is the difference in products? In future, we will see the cross-section of each obtained nanocarriers for drug delivery applications. Here we can see the different cross-section of drug carrier nanofibers using electrospinning approach. In a single fiber, a drug is dissolved or dispersed in a fiber matrix. In hollow fiber, the drug either dissolves in the fiber matrix or fill in the cavity of fiber. Core shell fiber apply two different fiber solutions and the drug is covered in the core of the fiber. In the genus fiber, two different bioactive agents dissolve or either disperse in the fiber, may be located side by side of the fibers. In the coaxial, the medicine is dispersed in the core and shells around it. Triaxial and multiaxial fiber is composed of more fiber solutions using different bioactive agents. Island in C is a multi-axial technique in which all above agents are embedded in the one fiber matrix. In this slide, we can see the synthetic polymers and natural polymers that are using for materials for electrospinning. Also, we can see the materials used for microfluid spinning are only synthetic polymers. However, we can see materials used for both electrospinning and microfluid spinning can be natural polymers and synthetic polymers as well. The main three approaches to fabricate drugs in spinning methods include electrospray nanoparticles as drug carriers, Electrospun nanofibers as drug carriers, and electrospray nanoparticles that incorporated into electrospun nanofibers as drug carriers. Here we can see the list of potential drug delivery applications of electrospun nanocarriers. In the electrospray method, the bioactive polymeric solution is sprayed and generates dry monodispersed nanodroplets due to the separation of polymer chains via the solvent system. The advantages of this method are high encapsulation efficiency and scalable reproducibility and synthesis. In electrospinning techniques, the drug fabricate in the form of fibers. The operation parameters, solutions properties, and environmental condition is impact on the final fiber features. Recently, the more advanced approach is developed by a combination of electrospun and electrospray techniques, which incorporated hydrophobic or hydrophilic electrospray drug nanobits in the electrospun fibers, while only a few studies are reported in this domain. Here you can see the list of potential drug delivery applications of electrospun nanocarriers. In the form of particles, particle in the fiber, and fibers. Three different approaches. In conclusion, it's clear that the electrospinning method, by generation of desired nanofibers and nanoparticles, possesses excellent potential in pharmaceutical applications. However, optimization of the different factors is crucial to generate nanocarriers with desired function and morphology for drug delivery application. Also, the electrospinning approach is an effective strategy for sustainable drug release, target delivery, and encapsulation of drugs. Moreover, there are drawbacks to this system for commercialization of nanocarriers for drug delivery. Therefore, further studies, standardization, and understanding of public health concerns are required to develop marketable products. Professor Siram Ramakrishna and I wish all of you have a successful conference. You are all welcome to contact us at the email address given in this slide. Thank you for listening.